with the USDA AIPL in Beltsville, and he will continue the story of hunting for QTL for calving traits. Please, John.
Thank you, Herman. I hope you don't think I'm too informal for wearing my little jacket up here, but it's like 4 degrees in this room and I'm freezing. Herman, I apologize for correcting you. We are no longer AIPL despite what my badge says. We have been reorganized and we are now the Animal Genomics and Improvement Laboratory. We've been combined with Kurt Van Tassel and Tad Sonstergaard and George Liu's group. So we're one big happy family now, which is probably fine because we were all co-authors anyway, as well as our colleague Tim Smith at the USDA Meat Animal Research Center in Clay Center, Nebraska. And all of my slides will be readily available on our website when I get home. So it might be easier to go ahead and download them than to try to grab photos here and there, or I'd be happy to give you a copy if you like. So as far as the overview of the talk, we're certainly not the only group working on chromosome 18. Donna Berry's group has been working on it. Other groups, the group in Scandinavia who had a talk earlier in the week. So I'm going to describe in a very brief way what we know about chromosome 18. I'm going to tell you how that led us to conclude that perhaps we need to do some sequencing. When we did sequence, since sequencing is magic now, I will explain what we learned when we looked at the data. What did we do when an analysis of those data revealed that we still had some challenges ahead, and I will then describe perhaps what we're going to do in the future. And I was really happy when I was able to go and find an actual photograph, a photograph from a karyotype of, I think that's a GR banded version of chromosome 18. So this is the guy we're dealing with here, although this is black and white, and I assume it's not that in real life. Now, these references listed are not meant to be a comprehensive list of every paper that's discussed chromosome 18. But there have been a number of reports indicating that there are QTL on chromosome 18 associated with dystocia, as well as conformation traits associated with body size and body shape. However, unlike some work, sort of a straightforward bioinformatic analysis using SNP data has not led to a simple explanation for what's happening there, possibly because we're dealing with complex phenotypes rather than, say, a more traditional Mendelian sort of trait. So we were successful in our group, and other groups in France and other places have been successful at using next-generation sequencing to identify causal variants. And Tad Sonstergaard made the mistake of saying the sequencer wasn't busy at the moment, so we remedied that problem. Now, chromosome 18 is different, and I'll explain what I mean. Our QTL on chromosome 18 has large effects on several traits, including traits associated with calving, dystocia, and stillbirth. In this case, the large effects are on the direct effects, but there's also an effect on the daughter or the maternal component, conformation, and then those also affect kind of efficiency traits in the sense that when you have increased dystocia and that sort of thing, you have reduced longevity and then reduced lifetime economic profitability. And perhaps the way I approach thinking about this is that when you have large calves born, you have increased dystocia, then you have reduced cow lifetime in the dam and decreased profitability. So this is something that has an impact on our production cattle population. And this is a figure that I copied out of our 2009 paper where we first reported our findings related to this. And you can see there are 8 traits here associated with our marker. And in fact, I showed the name here. I should replace that with the RSID because it now has one. And one of the questions is always, does the association hold over time? And Herman, I appreciate you showing one of our old Manhattan plots. We now have new, fancier Manhattan plots on our website. So you can grab one of those if you like. And we can see that generally speaking, across the 8 traits, we still see a large effect on chromosome 18 associated with this QTL. And the effects here are shown in terms of additive genetic standard deviations. rather than allele substitution effects. This is a heat map describing the correlation matrix among the 8 traits that we consider the conformation traits, the calving traits, net merit, and productive life. And the key thing here to note is the top value, the top figure, is showing you, it's a heat map of the correlations for the DGV calculated using all of the SNPs in our data set. The bottom figure is using DGV calculated using only the SNPs on chromosome 18. And you can see that there's a dramatically different correlation structure. And some of you might ask the question, well, in some sense, is this a significantly different pattern of correlation? And that's turned out, I would argue, yes, from a strictly heuristic approach of looking at it, but it's actually a little trickier problem than you might think to actually test these for differences. And we have a recent paper in Journal of Animal Breeding and Genetics talking about some ideas on dealing with correlation matrices. And while we did not look at gestation length in our work, Christian Malteca's group also reported that the same SNP has the largest effect of any SNP on the chip with respect to direct gestation length. So we saw large effects on the direct calving traits, and that's mirrored here. Now, if you look in the older paper, you'll see the coordinate for the key SNP has moved around a little bit as the assemblies have been refined. So it started out at 57, 125, 868, and it's moved around a little bit, same general region. It is intronic to a gene called Siglec12. Again, from the earlier paper, I believe it was in Siglec6. The assembly changes, and so these things change a little bit. And I've already described the effects on gestation length and birth weight. But I would like to point out that Perfield from Donald Berry's group did a really excellent paper this year, pulling together a lot of information about this complex and making a lot of information available in a very nice form. So if you want to get caught up, that's a good place to start. So where did this problem come from in the first place? This is Osborne-Dale-Ivanhoe. I don't know if this actually works. Osborne-Dale-Ivanhoe, and one of the things about Ivanhoe was that he was noted for his size as well as the large size of his daughters. And what you can see here, if I aim this in the right place, is this is chromosome 18, and this plot shows the haplotype. So this is the direct genetic value for each haplotype. And his maternal haplotype, which is in kind of the salmon color here, is not very good, whereas his paternal haplotype is basically breed average. Well, he was influential and had some important sons, including round oak ragapple elevation, who received that haplotype from his maternal grandsire, Ivanhoe. And in addition to having tens of thousands of daughters, Elevation had more than 2 million granddaughters. So this haplotype is spread all over the U.S. Holstein population, and it's also segregating in other black and white cattle populations. Now, one of the things we notice first when we look at this region, say in the NCBI viewer or in the whatever genome browser you care to use, is that there are lots of genes here. And in fact, on Tuesday, there was a talk proposing CCAM18 or mutations in that gene as possible causative mutations associated with this phenotype. And so the lower figure here is intended to show you that the SNP is in fact intronic to Siglec12. But the situation gets a little bit more complicated. Yali Hu, who worked for George Liu in Beltsville for a while, has also shown that there are several copy number variants in this region. There is a loss and a gain to the left, and as well as a gain to the right. Now, one of the things that can happen when you have a region like this with lots of copy number variations is it can be difficult to assemble. Now, I sort of hope Bob Schnabel's not here to ask me questions that will reveal my sort of ignorance of how this really works, but what if we look at a different trait? Because the story we told in our 2009 paper is this. In the mouse, Siglec6, I changed it to Siglec12 here to match the current assembly, sequesters leptin. from the circulatory system. This is associated, again, in the mouse with increased gestation length and then I would assume higher pup birth weight. Now, the comparison can't be perfect because, the mouse is polycotomous and has different placentation and all of these things, but it's an interesting story. So what would this tell us? Well, it says we should look at calf birth weight to see if that's higher as well, related to this increase in gestation length. Because, of course, you would expect very large calves at birth to be associated with greater dystocia. And when you have greater dystocia, you're also then at higher risk for stillbirth. Particularly in the U.S., the definition of stillbirth includes calves born dead or calves that die within 48 hours of birth. So a calf that was born with great difficulty and later dies would count in the stillbirth. Unfortunately, in the U.S., we don't have birth weight data routinely recorded, and what is recorded on farms doesn't usually make its way to the national database. And some of you may suspect collusion between the chairman and myself, but 4 years ago at the last World Congress, Herman and I got together and agreed to do a project to try to sort this out, because in the eastern part of Germany there are large, intensively managed dairies that are similar in many respects to U.S. dairies, and they also use some U.S. genetics. And they have extremely comprehensive data recording, these cooperator herds. And so we thought, hey, this is a way we could approach this. So we developed a selection index-based predictor of calf birth weight, well, a PTA for calf birth weight, and then we did some GWAS and gene set enrichment analysis, and that's in Journal of Dairy Science. Well, fortunately, I would say for us, there is one of the larger effects in the genome is located in the same linkage block as our SNP on chromosome 18. I thought that was encouraging. But you always want to ask yourself, are we measuring anything new? And what I mean by that is most of the predictors in the birth weight index already had large effects for this marker. So maybe all you're really doing is identifying things you would have identified without the birth weight predictor. However, one of the things we did get to here is LHX4 on chromosome 18, which has been associated with cow body weight and body length, so associated with larger sort of frame and skeletal size. As noted, there were 4 SNPs on chromosome 18, and some other markers with large effects, like I believe there's one on chromosome 26, for example. They were intronic or they were adjacent to genes that don't really have much annotation with them, so we don't know what their function is, particularly what their function is in the cow. So we're still running up against that challenge in the cow of not always having good information about the genes. We also did some simple gene set enrichment analysis and said, can we identify any interesting pathways? And I put this up here because we identified regulation of actin cytoskeleton as a pathway. Well, I'm sort of skeptical about this because I feel that one of the questions we always should ask ourselves is there a plausible association, not just a statistical association, but sort of a mechanistic association between things like these pathways or genes we find and the actual phenotype we observe. So it's unclear to me. You could sort of hand wave and say, well, of course, actin is going to be associated with growth and development, but we need more data about how that would actually work before I'd feel really comfortable. So that was sort of a traditional SNP-based association type study, and it didn't really get us to any clear answers. So our next step was to do some sequencing. And on your right, you'll see a pedigree-based design that I came up with to sequence several bulls. I actually started with Arlinda Rotate because he had sons with high reliabilities for sire calving ease and also large differences. So you'll see Melwood and Jed have a difference of 7 points between them for sire calving ease. Breed average is about 8. The larger the number, the less desirable because that's a higher incidence of dystocia. And so then I grabbed some maternal grandsires and some other sons. And then my colleague Paul Van Raden said, well, that's fine as far as that goes, but if what you care about is the haplotype, why don't you just sequence the haplotypes you're interested in? So on your left, you'll see that we did that for 3 bulls, including an animal that's homozygous for our tag SNP, because it's not like a Mendelian recessive, so we have a couple of hundred homozygotes in the population, and so that's Wedgwood Laramie, a Canadian show type bull with some difficulties with sire calving ease. And because he shows up in a lot of the pedigrees, we had hoped to sequence Paclomar combination, but his DNA has been difficult to locate. So how many scientists does it actually take to do the sequencing? Well, hopefully on Tuesday you went to Tabitha Cooper's poster. Derek just gave a talk, and hopefully he's in here to bail me out on the question time. And then my technician, Dan Null, had to stay home and actually do some work so I could come up here. We ran this on our Illumina HiSeq that we have in Beltsville. We did all of our DNA extractions from semen, some of which was taken from the CDDR, some of which was purchased commercially. And we got fairly good depth of coverage for most bulls except Lystel Leduc, who was a little under 8x. We had some issues. I think that was we had a run where the sequencer had problems at the paired-in turnaround, and so we got less depth than we hoped for. So we took the data off of the sequencer, and with Derek's help, we did paired-end read alignments and split read mapping. And one of the interesting things we found is that there appears to be a tandem duplicate of 2 exons and an intron within the IG-like protein domain in Siglec12. In addition, some of the animals that were heterozygous, so they carry a major and minor allele for the tag SNP and have desirable PTA for sire calving ease, also had a deletion near the end of the protein. And we showed this in our poster at PAG. I'm going to focus on the bottommost panel, the line graph there, which is just showing the read depth. Now, one of the things you notice is all of a sudden we have a huge pile of a bunch of reads piled up there, and this could represent a GC bias in that region of the genome. That's a bias in the Illumina chemistry, or it could represent problems with the assembly. Perhaps I should clarify that. It could represent problems with the assembly because it's sometimes difficult to assemble regions where you have repetitive DNA. And so in some assemblies, the assembler will combine 2 contigs that are similar. The problem is sometimes they actually belong on top of each other, and sometimes they belong adjacent to each other. And this is the part that I hope Bob isn't here to see. This is my extremely simplistic explanation of what's going on with assembly. So we have short, particularly with Illumina, we've got short reads, and we want to assemble those into longer pieces of DNA that we eventually assemble into, say, a chromosome. And this is a computational process. It's not a deterministic thing where you get a single definitively correct answer. And the Liu paper I reference here refers to the current BTAU4 assembly, and then the Zeman paper describes the University of Maryland 3.1 assembly. So as I said, it's imperfect. And this is an attempt to explain that sometimes a region where there's a single element should actually have elements next to each other. And that could explain this pileup we have on the read depth. So what this means is we weren't really sure if this was a correct result or not. It could be a genuine biological phenomenon, or it could reflect an error in the current reference assembly. So we decided, I went to PAG in January, and I learned at PAG that the greatest thing since sliced bread is PacBio sequencing. Our colleague Tim Smith has a PacBio sequencer. And the key here is that when you sequence DNA with PacBio, you get long reads. Okay, so instead of getting 100 base pairs of sequence, you can get very long pieces of sequence. So what we did is we went into the back libraries we have in Beltsville. Okay, we went and took Domino's DNA, and we pulled out four contigs from the back that overlap with each other, and they tile across the region that we're interested in here on chromosome 18. So our idea here is that we would do long read sequencing on them, and then we would be able to construct a new local assembly and then align our Illumina data against that to try to resolve the issue of, is the tandem duplicate real or not? And so the reads were processed. We extracted the DNA in Beltsville did some cleaning to try and take out the vector DNA, and that was sent to Clay Center, USDA Mark. Unfortunately, at Mark, they had to pull all of the back DNA we sent them to have enough material to construct a sequencing library. And some of you are probably thinking that doesn't sound right, and I'll discuss in a minute what the problem with that is. The read, it was just then run on the instrument. Reads were assembled into contigs using smart analysis. Tim Smith did this for us, and he sent us back 44 contigs with an N50 of 31 kb. So we then took the PacBio contigs and we aligned them against the UMD 3.1 assembly with Mummer. And the idea there was to say, does our new assembly using the PacBio data, how similar or discordant is it with the current reference assembly? We then aligned our short Illumina reads against the PacBio contigs using BWA. And Derek Bighart did some paired in discordancy mapping using custom software that he's written, and I think he may have discussed in his talk. And I'm going to show a few of these Mummer plots, and the point is, in an ideal world, you have a line with a slope of 1 that says that the sequence in the PacBio contigs is a perfect match for the sequence in the reference assembly contigs. And for the discordancy analysis that Derek did, we only kept reads that were longer than 4 standard deviations above the mean or shorter than 4 standard deviations above the mean. And when you have those fragments, you're doing things like looking at segments of DNA where there are insertions or deletions relative to the assembly. And if you have regions where you have lots of discordant reads, this can indicate problems in the PacBio assembly. Certainly is possible. It could indicate the presence of repetitive elements, and I'm not sure that it's very likely. I was told it's not likely, but it's possible that there are structural differences between the Holstein cow and Herefords. And of course, Domino, which is the reference genome, is a Hereford. And we got very excited, or at least I got very excited when I first saw this, because this is a region on chromosome 18. Our SNP is contained in the blue segment down at the bottom left. There's a big stretch in the middle of our contig that includes DNA that does not map to the reference assembly. And then up there, sort of in the middle in the top in blue, you have sequence on the end of ours that anchors back to the reference assembly. So I thought, hey, this is exciting. We found new DNA. So we have this 10 kb of new DNA, except that it was vector DNA. And you'll recall that I said earlier that we tried to process this out before we sent it to Clay Center, and we were not successful. We don't do a lot of work extracting the DNA, like say from the back libraries, and I think we're going to have to pay more attention to that. We usually work with semen and constructing Illumina libraries from DNA taken out of semen. So we had some issues there. But there are clearly assembly problems in this region. So what you see in these red segments that are boxed in, that's duplicate sequence. And there's also more duplicate sequence downstream on the UMD 3.1 assembly relative to the PacBio assembly. And I know that those of you that are laboratory biologists, rather than sort of, I think of myself as more a traditional animal breeder, is that this is a much more complex analysis than using SNP genotyping. The SNP genotyping technology is mature. It's relatively foolproof. It's probably as foolproof as any laboratory technique can be. This is not the case, and unsuccessful experiments are to be expected. In particular, we did not produce a sufficient quantity of high-quality DNA for Clay Center to construct the PacBio libraries properly. When they pulled the DNA to get enough of it, this was a problem because our backs overlap. So you're now pulling overlapping DNA, sequencing it, and trying to assemble it. And that's, PacBio will tell you not to do that, and people who know more about sequencing than I do tell you not to do that. So, Tim did it because he had the material that we sent him, and we figured we'd see what we could find. And then data editing and error correction are critical. Again, we didn't get all the vector DNA out of the backs like we needed to, and we didn't have sufficient quantity of DNA. So what do we do now? And then on the next slide, I'll tell you where we actually really are at this point. We're going to try to reassemble the raw PacBio reads after we do some more quality control and data cleaning. I don't know that we're going to get substantially different results, but the data are available, so we should look at them. Next, we're going to resequence the back libraries. And we're also looking into getting the Chori libraries, which are from the Holstein calf that Monsanto prepared backs from. And this will help us get at the question, a better job of answering the question of, are there structural differences between Holsteins and Herefords in this region? So we feel comfortable that there are structural variants in and around the Siglec12 gene that are associated with differences in sire calving ease, as well as the conformation traits and stillbirth. We also think some of the SNPs are misplaced. The third point about the 8 kb region downstream of our SNP that appears to be misassembled, I showed you those data in the Mummer plot, but the causal variant has not yet been conclusively identified, and unfortunately we were not even able to answer our question of, is this tandem duplicate we observed in our earlier data, is that real, or is it still an artifact related to an error in the assembly? So that's certainly, this is our first foray into working with the PacBio instrument and preparing materials to go on there. And obviously we have a little bit of work to do because it's a different sort of process than working with semen and you get lots of nice DNA fairly easily out of semen. It's not so the case of taking the backs out of the bacterial libraries. But I would certainly like to acknowledge the help of several technicians, Ruben and Sasho in Beltsville, as well as Renee and Clay Center, because they did what we told them to do in preparing samples and shipping samples. If I didn't tell them what to do correctly, they can't be blamed for the results. I would like to acknowledge our appropriated funding from USDA, as well as the cooperation of the Council on Dairy Cattle Breeding and the Cooperative Dairy DNA Repository for providing data as well as DNA. And I would be happy to answer any questions you might have.
Thank you, John. Please, any questions? Yes, please, sister.
Could you comment on the usefulness of PacBio using the error rate of sequencing? So while PacBio is quite long read, and previously it's known for quite high error rate, so if you align these reads and look for duplication, there might be alignment problems.
Krista, I'll tell you the little I know, but I have to admit that I'm not an expert. But there was a tremendous amount of discussion of the error rate issue at PAG. And it's my understanding that a combination of new chemistry for the RS2 as well as improved software for doing error correction, that the error rate is much lower. I mean, certainly it was 15 or 18% on the old chemistry, and it's probably not as good as Illumina now, but I think it's substantially improved, particularly if we were to do multiple runs on the instrument. But I do agree with you. If you're not careful with those data, you could still have artifacts in PacBio that are a problem with the PacBio and not necessarily a problem with the Sanger sequence from the reference.
Any other questions? Yes, please.
So, John, we haven't met, and this absolves me from looking like this is a plant question. I like the story that your presentation gives. Do you think that PacBio has a need in, say, assembly reconstruction? Or do you think that it's just a tool that we use for this purpose?
Well, to the person I don't know in the audience, my colleague Dr. Bickhart here. So no, I think the tool is useful. I think the problem we have here is that unfortunately we didn't provide the proper inputs to the tool. And perhaps to get back to Krista's question as well, one approach here might be to do both PacBio sequencing to get long reads, and then to use the Illumina long read service, so that you would have long reads from two different sort of techniques and chemistries across the same region, and then you could perhaps try to do sort of a consensus alignment to construct maybe a most putatively true assembly in that region, and we could then align our short reads against that consensus assembly. Some of you might be wondering why we didn't go to Illumina to get in long read, and my recollection at the time is we were trying to get preliminary data for a grant application, and it was something like a 6-month wait in the queue for the Illumina long read. So we took advantage of resources we had available at the time.
Yes, please.
I have a little practical question. With this zone or region on chromosome 18 that's associated with dystopia, there are SNPs that we're using for our genomic evaluations around the world and in the U.S.
Yeah.
Should you have expected higher gains in accuracy for the dystocia-related traits or not? Or is there a way to gain in accuracy for those traits compared to other traits? Or is there a way to continue to improve that by selecting different traits for gene affecting networks?
Well, I mean that's a good question, and when you ask a question about gain in reliability, or if we want to go backwards, in the words of one of my esteemed colleagues, to look at gain in accuracy, part of the issue is we have seen lower gains than expected for the calving traits. And we've also had some challenges with our interval trend validation for the calving traits from the U.S. And we have spent a substantial amount of time fitting different models, first versus later parities. We've tried fitting linear models, sort of linearizing, probit and Snell scoring type approaches. And we still don't have a good explanation for what's going on with our calving traits. Like, our rank correlations are pretty good when you look at our PTAs compared to other interval participants. But there does appear to be something a little flaky in those evaluations, in the sense that we don't gain as much accuracy from adding the SNPs as you would expect. And maybe one other thing to get that might help explain it as well is this is one of the largest effects that we've seen in the genome, but my recollection is that it accounts for maybe 15% of a genetic standard deviation, say for sire calving ease. So relative to the other effects, it's very large. But if you put that in terms of either the proportion of additive genetic variants or proportion of phenotypic variants explained, it's actually not not huge relative to, say, the traditional QTL definition of 30% of variants explained.
I think I'd take one last question. Sophie, please.
Did you see any association of this region with maternal cavities that might back up the negative correlation that we keep finding in this way?
My recollection is it was one of the larger effects for maternal calving ease, but the magnitude of the effect was fairly small. It was not certainly not nearly as large as it was for sire calving ease. So.
Yeah, I think we should probably not stretch it too far. And, I have to thank, first of all, Peter and John for giving 2 excellent talks. And as we just had a lively discussion, thank you for that. And I would like to thank all the people who participated and all the people who attended. Thank you.